Hello everyone, welcome to the channel Microbial World. In this video, we will be studying about type II hypersensitivity reaction. Type II hypersensitivity reaction is an antibody-mediated reaction where the immune system mistakenly attacks self-cells or foreign cells with the help of antibodies such as IgG or IgM. This will lead to cell destruction, inflammation or dysfunction. Let us look into the mechanism of type II hypersensitivity. Antigen recognition. The immune system recognizes an antigen as harmful. These antigens could be intrinsic antigen or extrinsic antigen. Intrinsic antigens are self-antigens that is present on the cell membrane. For example, normal cell proteins. Extrinsic antigens are foreign antigens that attach to the cell surface from an external source, for example, drugs or pathogens. Once the antigen is recognized, antibodies such as IgG or IgM antibodies will bind to the antigen on the target cell, and this will lead to cell destruction. As type II hypersensitivity reaction is mediated by antibodies and resulting in cellular destruction, it is also called as antibody-mediated cytotoxic hypersensitivity. Now the cell destruction occurs through these following mechanisms. Complement-mediated cell lysis or classical pathway activation, opsonization and phagocytosis, antibody-dependent cellular cytotoxicity, and antibody-mediated cellular dysfunction. Complement-mediated cell lysis. This pathway will lead to direct cell destruction through the activation of complement system. Now let's understand how this mechanism occurs. IgG or IgM antibodies bind to the antigen on the target cell surface. C1 complex, which is made up of three subunits C1q, C1r and C1s, will bind to the antibody Fc region. This activates classical complement cascade, that is complement proteins C2, C3, C4 and C5. Let's see how this activation occurs. When the C1 complex binds to the antibodies on the pathogen, it activates the complement protein C1s. C1s will act as a scissors to cleave other complement proteins. C1s cleaves complement protein C4 into C4a and C4b, and it also cleaves complement protein C2 to C2a and C2b. Then C4b and C2a together forms C3 convertase, an enzyme which will amplify the process. C3 convertase will cleave the complement protein C3 into C3a and C3b. C3b then joins C3 convertase to form C5 convertase, and C5 convertase will split the complement protein C5 into C5a and C5b. Then finally the complement proteins C5b, C6, C7, C8 and C9 together forms cylindrical membrane attack complex. The membrane attack complex will create pore in the cell membrane, causing cell lysis and death. Hemolytic transfusion reaction is an example of this type of mechanism. Opsonization and phagocytosis. This pathway leads to cell destruction by phagocytosis. Antibody IgG coats the target cell acting as an opsonin, which means a marker for destruction that enhances the process of phagocytosis. Complement protein C3b binds to the antibody-coated cell, enhancing opsonization. Macrophages and neutrophils have Fc receptor and C3b receptor. These receptors recognize the antibody IgG and complement protein C3b, leading to phagocytosis. Immune cells engulf the opsonized target. Lysosomal enzymes digest the cell, causing cell destruction. Autoimmune hemolytic anemia and immune thrombocytopenia follows this mechanism. Antibody-dependent cellular cytotoxicity. This pathway leads to cell death through natural killer cells and cytotoxic molecules. IgG antibodies bind to the antigen on the cell surface. Natural killer cells express Fc gamma R3 receptors, that is CD16 receptors, which bind to the Fc region of IgG. Once the Fc receptor on natural killer cells binds to the antibody, natural killer cells will release perforins, that is pore-forming proteins, and granzymes, which is proteases. Granzymes activate caspases, leading to apoptosis, that is programmed cell death. Graft rejection and viral infected cell destruction is based on this mechanism. Antibody-mediated cellular dysfunction. In some cases, antibodies do not destroy the cell, but they alter the function of the cell by blocking or stimulating receptors. The diseases myasthenia gravis and Graves' disease follows this mechanism. The ligand acetylcholine normally binds to acetylcholine receptor at the neuromuscular junction. But autoantibodies target and block these receptors, preventing acetylcholine from binding. So the antibodies are inhibiting the binding of acetylcholine to acetylcholine receptor. This leads to muscle weakness. The ligand thyroid stimulating hormone normally binds to thyroid stimulating hormone receptor on thyroid cells. But in Graves' disease, autoantibodies called thyroid stimulating immunoglobulins will bind to the thyroid stimulating hormone receptor and stimulate the receptor, causing excessive thyroid hormone production. This is called as hyperthyroidism. So this is all about type 2 hypersensitivity reaction. Thank you.